Cherokee Nation for taking a DNA test that's not allowed anymore. Meanwhile, ahead of issuing the party's response to the State of the Union address, the 2018 Georgia gubernatorial candidate Stacey Abrams.
Whispers say the Cherokee carry ancient bloodlines that do not fit the textbook story. Is there a buried chapter hidden in DNA that America refuses to read? Are Old World markers really found among Cherokee families? Or are we chasing rumors and results taken out of context?
Tonight, the leaders of the Cherokee Nation are speaking out against Senator Elizabeth Warren's campaign video touting her Native American ancestry courtesy of a newly revealed DNA test. The tribe saying in part, quote,
"And who decides identity, labs, or nations? We'll unpack sensational claims and show what peer-reviewed genetics and Cherokee leaders actually say. We'll test the darkest secret against records, migrations, and politics, then map what is known, what is disputed, and what remains a mystery. What DNA really decides." When people hear about DNA testing, they often imagine that a simple cheek swab can answer the biggest questions of identity. For the Cherokee Nation and other Native nations, however, DNA has never been the key to belonging. Cherokee leaders have repeatedly explained that tribal citizenship is not something a lab can decide. In 2017, the Cherokee Nation's Secretary of State issued a blunt statement. A DNA test is useless to determine tribal citizenship. That line is more than just a political stance. It reflects centuries of law, tradition, and sovereignty. To belong to the Cherokee people is not about percentages of blood or genetic markers. It is about being part of a living community, governed by its own rules, treaties, and continuity as a nation. This difference between citizenship and ancestry is crucial. A DNA test may reveal markers that connect someone to broad populations across continents, but it cannot place them inside the political body of the Cherokee Nation. Citizenship is defined by each tribe's constitution and laws, not by a database of haplogroups. For example, the Cherokee Nation requires lineal descent from an ancestor listed on the Dawes rolls of the early 20th century. This is a matter of record, genealogy, and law, not genetics. DNA cannot override the decisions of sovereign governments that continue to exist today. By confusing ancestry with citizenship, people risk misunderstanding both the authority of Native nations and the cultural fabric that keeps them alive. Even when it comes to ancestry, genetics offers only part of the picture. As Teen Vogue reported in a widely shared article on Indigenous identity, DNA tests cannot measure culture, kinship, or the lived experiences that define belonging. Many federally recognized tribes across the United States have openly rejected DNA as a tool for enrollment, stressing instead that being Native is about relationships, traditions, and responsibilities. One can carry a haplogroup marker without ever having spoken the Cherokee language, attended a stomp dance, or participated in community life. Conversely, a person raised in Cherokee communities, steeped in language and ceremony, may not have what a commercial DNA kit would flag as Native markers. The danger is clear. Over-reliance on DNA risks flattening a living culture into little more than a test result. The broader issue is how framing DNA as destiny can erase nations themselves. Time magazine has highlighted the political stakes of treating genetic ancestry as proof of identity. When outsiders insist that DNA can prove indigeneity, they undermine tribal sovereignty and the right of Native peoples to define themselves. It shifts power away from Native governments and into the hands of laboratories and corporations. It also feeds into old colonial ideas that Indigenous identity is something to be measured, quantified, or validated by outside authorities. For the Cherokee, whose history includes forced removal, the Trail of Tears, and generations of battles to protect their nationhood, this is more than an academic issue. It is a fight to preserve self-determination. The truth is that DNA can be a useful tool for studying human history and migration, but it was never meant to replace political belonging or cultural continuity. The Cherokee Nation makes this clear. They are not defined by test tubes, but by peoplehood. Citizenship rests in law. Culture is passed down through ceremony, story, and kinship. Identity is lived daily, not extracted from saliva. To confuse these domains is to miss the heart of what it means to be Cherokee. If DNA cannot decide citizenship, what can it reveal about origins, migrations, and the deeper story of the Cherokee people? The textbook origin story. For decades, the story of Native American origins seemed clear, even settled. Geneticists studying mitochondrial DNA, the genetic material passed down the maternal line, built a model that has dominated textbooks and classrooms alike. According to this model, nearly all Native Americans descend from 4 maternal haplogroups, labeled A, B, C, and D. These lineages are thought to have emerged in Asia tens of thousands of years ago, carried by small groups of people who crossed a land bridge from Siberia into Alaska during the last ice age. This land bridge, called Beringia, existed when sea levels were lower, forming a frozen corridor that connected 2 continents. Once those early people arrived, they spread southward into the Americas, eventually giving rise to hundreds of distinct nations and cultures. In this telling, the Cherokee, like all other native peoples, are rooted in the same migratory wave from Asia. The strength of this model lies in its simplicity. A, B, C, and D appear again and again in native samples, whether from ancient remains or living descendants. PubMed-indexed studies over the years reinforce the idea that these 4 haplogroups serve as a kind of genetic signature of native ancestry. To many scientists, this meant the debate was closed. If nearly everyone sampled fit one of these categories, then the conclusion was straightforward. All Native Americans share the same Asian origin. The land bridge theory wasn't just plausible, it was stitched into the very fabric of human genetics. But science has a way of complicating neat stories. By the late 1990s, researchers began to notice something odd. A 5th haplogroup, labeled X, was present in certain Native American groups. Its frequency was low compared to A through D, but it appeared consistently enough to demand attention. This wasn't a random anomaly, it was a real signal. Suddenly, the model of 4 founding mothers had to be updated. Peer-reviewed papers in both PubMed and Science Direct documented X in populations like the Ojibwe in the Great Lakes and some Algonquian-speaking groups in the Northeast. While the Cherokee were not always directly sampled in these early studies, the discovery of X suggested that the genetic roots of Native America were more varied than scientists had believed. The finding of haplogroup X added fuel to a growing fire. Unlike the 4 classic haplogroups, X was not common in East Asia. Instead, it showed stronger ties to populations in Europe and the Near East. At first glance, this raised provocative questions. Could haplogroup X in North America point to ancient contact with Europe long before Columbus? For a moment, some popular accounts embraced that narrative. But geneticists cautioned against jumping to conclusions. The presence of X didn't necessarily mean Europeans had crossed the Atlantic in prehistoric times. Later work provided crucial context. Studies from institutions like California State University, East Bay, showed that haplogroups also existed in parts of Siberia. That discovery reframed the puzzle. If X was present in Asia, then it could have traveled the same Beringian route as A through D. In other words, its presence in North America did not demand a transatlantic explanation. It could still fit within the framework of Ice Age migrations, albeit as a rarer lineage carried by fewer families. The once shocking presence of X was therefore absorbed into the broader model, expanding it without overturning it. Today, the mainstream genetic consensus still points to multiple waves of migration from Asia into the Americas, with haplogroups A through D as the primary lines and X as a recognized, though minor, addition. This framework explains the overwhelming majority of Native American maternal lineages, including those found among Cherokee descendants. Yet the discovery of X also opened the door for speculation, because its rarity and unusual distribution leave room for questions. Could it hint at forgotten migrations? Could it be a remnant of contact we don't fully understand? And this is where the Cherokee enter the spotlight. While mainstream genetics remains cautious, some researchers and writers argue that Cherokee DNA includes not just X, but also lineages far outside the expected pattern. They claim to see traces of the Mediterranean, the Middle East, and even Jewish ancestry embedded in Cherokee families. These claims defy the neat boundaries of the textbook model, suggesting that something extraordinary, something unrecorded in conventional history, may have happened. So where do these shocking claims of Old World Cherokee DNA come from? The claims that lit the fuse. If the mainstream genetic model sketches a cautious, structured story of Native American migrations, the alternative claims about Cherokee DNA ignite something far more explosive. Much of the firestorm can be traced back to the work of Donald Yates and DNA consultants, who published books and reports arguing that the Cherokee carry mitochondrial haplogroups far outside the expected A, B, C, D, and X framework. According to their studies, Cherokee descendants sometimes showed T, U, J, and H, lineages typically associated with Europe, the Middle East, and North Africa. In their interpretation, these were not late arrivals from colonial marriages, but echoes of ancient contact, proof that the Cherokee had ancestral ties stretching across the Atlantic to Mediterranean and even Jewish populations. It was a dramatic suggestion, and one guaranteed to make headlines. After all, if true, it would mean that American prehistory was entangled with Old World civilizations long before Columbus arrived. The boldness of these claims ensured that they spread quickly. Articles citing DNA consultants framed the Cherokee not only as an indigenous nation of North America, but as a people with bloodlines that connected them to Egyptians, Berbers, and Hebrews. In one report, Yates argued that haplogroup T appeared with such frequency in Cherokee-descended samples that it could not be dismissed as chance or recent admixture. U and J, too, cropped up in numbers that seemed, to his team, far beyond what colonial history could explain. By tying these results to Mediterranean and Jewish populations, Yates painted a picture of the Cherokee as guardians of a hidden legacy, one that mainstream science had overlooked or even suppressed. For many readers, the story was irresistible, and when Elvis Presley's name entered the conversation, the theory gained another layer of pop culture appeal. Presley, who publicly embraced both his Jewish and Cherokee roots, was said to carry mitochondrial haplogroup B, a lineage firmly considered Native American. Yet, family lore and genealogical records also tied his maternal line to Jewish ancestors. Yates and others seized on this as a living example of how Cherokee and Mediterranean markers could intertwine in one person. It seemed to provide a famous face for the controversial narrative, the king of rock and roll himself, embodying the anomalous DNA mystery. But here, the ground grows shaky. Celebrity ancestry claims often rest on rare samples, unverifiable sources, or selective interpretation. In Presley's case, much of the evidence comes from genealogical reconstructions and isolated DNA reports rather than broad, transparent studies. It makes for compelling storytelling, but not necessarily strong science. Critics argue that spotlighting Elvis as proof of unusual Cherokee DNA risks confusing anecdote with evidence. A single individual cannot carry the weight of rewriting an entire population's history. This raises the larger issue, the methods and assumptions behind these alternative claims. Many scholars have pointed out that studies like those promoted by DNA consultants rely on small, self-selected samples, often drawn from families who already believed they carried Cherokee ancestry. That approach can skew results and inflate the presence of unexpected haplogroups. Critics also stress that post-1492 admixture is a simpler explanation for many of the European-coded haplogroups reported. After centuries of intermarriage between native women and European, African, and Jewish settlers in the Southeast, it is hardly surprising that descendants today might show diverse genetic markers. To leap from those findings to ancient Mediterranean voyages, critics say, is to bypass the more straightforward historical record. Academic journals, such as those published through Taylor and Francis Online, have called for caution when interpreting anomalous DNA findings. Population genetics is complex, and mitochondrial haplogroups only reflect one narrow slice of ancestry, the maternal line. To claim they rewrite entire migration narratives requires extraordinary evidence, far beyond what small-scale studies have presented. Yet, the allure of a hidden Old World connection remains strong. It speaks to a deep human fascination with lost civilizations and suppressed histories, and the Cherokee DNA debate has become a lightning rod for those yearnings. And so, the mystery deepens. Are haplogroups like T, U, and J really evidence of forgotten transatlantic journeys, or do they simply reflect the tangled history of colonial admixture? Are we glimpsing the fingerprints of Phoenician traders, or just misreading the map of post-contact marriages? Could any of these markers point to contact before Columbus, or are we misreading the map? Pre-Columbian contact, or later mixing? When geneticists first detected haplogroup X among certain Native American groups, the reaction was electric. Unlike the four traditional native haplogroups, A, B, C, and D, X was rare and geographically puzzling. It did not appear in high frequencies in East Asia, the assumed source of all native ancestry. Instead, it was found in Europe and the Near East. This opened the door for dramatic theories. Perhaps Europeans, Phoenicians, or even ancient Israelites had crossed the Atlantic thousands of years before Columbus, leaving their genetic signature among the Cherokee and other native peoples. For a time, this possibility captured imaginations both in academic circles and in popular culture. But science thrives on new data, and broader datasets quickly reframed the debate. PubMed-listed studies, along with work from California State University, East Bay, showed that haplogroup X was not uniquely European. It also appeared in Siberia, including among populations that could have migrated through Beringia during the Ice Age. Archaeogenetic evidence from prehistoric remains in North America further supported the idea that X was already present on this continent thousands of years ago, long before Europeans sailed west. In other words, X did not require a transatlantic explanation. It could be accounted for by the same migration pathways that brought A, B, C, and D. The most exciting Old World signal, once thought to be proof of contact across the ocean, fit just as well within the established framework of Ice Age dispersals. This pattern isn't unique to haplogroup X. Other lineages long associated with Native America also show signs of deep diversification within the continent. For example, peer-reviewed studies of mitogenomes, full mitochondrial DNA sequences, have traced sublineages such as B2 back to North America roughly 11,000 to 13,000 years ago. Published through repositories like PMC, these analyses reinforce the idea that native haplogroups didn't just arrive here, they evolved and branched within the Americas over millennia. The Cherokee and their neighbors are heirs to this deep, continent-spanning history, rooted in timeframes that far exceed colonial contact or European exploration. These findings underscore the resilience of the Beringian model, which continues to explain the bulk of genetic diversity among indigenous peoples. Still, not all unusual markers in Cherokee descendants can be explained by Ice Age migrations. Some haplogroups labeled European do appear, but they likely have more recent origins. History itself offers a straightforward explanation. Centuries of intermarriage in the Southeast. From the 1600s onward, European traders, settlers, and missionaries entered Cherokee homelands. Many of them married Cherokee women, both as personal unions and as political alliances. Enslaved Africans and later freed people also became part of Cherokee communities, further adding to genetic diversity. As Wired and other outlets have reported, genetics alone cannot timestamp when a haplogroup entered a lineage. A marker that looks Mediterranean in origin might reflect an ancient migration, or it could simply record a marriage that happened 2 or 3 centuries ago. Without archaeological samples tied to secure dates, the DNA cannot tell the whole story. This is where the science meets its limits. Haplogroups are powerful clues, but they are not smoking guns. They describe patterns of relatedness, not precise events in history. They cannot distinguish whether a lineage appeared in Cherokee families 1,000 years ago or in the era of transatlantic trade. That ambiguity leaves room for speculation and for competing narratives to flourish. One side leans on the dream of forgotten voyages, the other on the well-documented record of colonial admixture. The truth may contain threads of both, but without clear ancient samples, caution is the wiser path. And yet, even if the Cherokee DNA mystery does not prove lost civilizations, it still raises profound questions. Because if history already documents centuries of deep mixing, what can those records and the patterns they reveal tell us about who married whom and when? Trade, marriage, and the record trail. To understand the presence of unexpected DNA markers among Cherokee descendants, one need not turn to lost civilizations or ancient transatlantic voyages. The answers can often be found in the written record of colonial America itself. From the 1600s through the 1800s, intermarriage between native women and European men was not only common, it was a structural feature of life in the Southeast. Traders, soldiers, settlers frequently married Cherokee women, creating alliances that were both personal and political. These unions forged family networks that persisted for generations, linking native clans with newcomers in ways that changed both communities. Wired has reported on the impact of these alliances, showing how such marriages left long-lasting imprints on Cherokee family trees. A single union between a Cherokee woman and an Irish, Scottish, Portuguese, or Jewish trader could introduce a new mitochondrial DNA lineage into the community. Because mitochondrial DNA passes strictly from mother to child, that lineage would then persist in all of her descendants, generation after generation. Unlike autosomal DNA, which dilutes over time, mitochondrial DNA remains intact, a near-perfect genetic signature of maternal ancestry. In practice, this means that even one such marriage in the 1600s could explain the presence of a European-coded haplogroup in Cherokee descendants today. This leads to a critical limitation of genetic interpretation. As researchers on ResearchGate have emphasized, itself prove when a lineage entered a population. It cannot distinguish between a marker that arrived 10,000 years ago and one that entered just 300 years ago. Without securely dated ancient samples, the signal remains ambiguous. In the Cherokee case, much of the European-associated mitochondrial DNA almost certainly reflects the historical record of intermarriage in the colonial period rather than prehistoric voyages across the Atlantic. The genealogical evidence aligns with this. Names of European traders repeatedly appear in Cherokee family histories, and many were remembered precisely because of the influence their unions had on trade, politics, and land agreements. Complicating matters further is the way DNA is discussed in popular culture. Blogs and websites often present sweeping lists of native haplogroups, suggesting that anyone who tests into A, B, C, D, or X must carry native ancestry. But as genetic genealogist Roberta Estes and others have explained on DNA Explained, this is a dangerous oversimplification. Within those haplogroups are countless subclades, only some of which are truly Native American. Others are found exclusively in Asia, Europe, or Africa. Without specifying the subclade, it is misleading to equate a haplogroup label with native identity. This sloppy labeling has fueled confusion and, in some cases, outright exploitation, with DNA companies selling identity shortcuts that ignore both science and sovereignty. The record trail shows a much more grounded story. By the 1700s, traders like Cornelius Dougherty, Richard Paris, and others had taken Cherokee wives. Their descendants, documented in both tribal and colonial records, carried the blended heritage of two worlds. These families became cultural bridges, but they also introduced maternal lines that outsiders later interpreted as anomalies. What genetics reveals today is simply the echo of those historic marriages. The DNA does not lie, but it can be misunderstood when stripped of its historical context. And this misunderstanding matters, because when people equate haplogroups with native identity, they risk reducing entire nations to test results, while ignoring the lived histories of adaptation, survival, and cultural continuity. That erasure is not just an academic error. It has political and social consequences that ripple into the present. Beyond biology, why is this debate so charged? And who gets hurt when DNA is treated as a shortcut? Power, identity, and the backlash. Every few years, a public controversy thrusts native DNA into the national spotlight. The most famous came in 2018, when a political figure released results from a commercial DNA test to support claims of native ancestry. The story dominated headlines, but for native nations, it was déjà vu. The Cherokee Nation, along with other tribes, responded bluntly. DNA tests cannot prove native identity or citizenship. PBS reported that tribal leaders stressed a long-standing principle: citizenship is a matter of law, not biology. Genetic results may tell someone they share markers with broad populations, but they cannot capture the responsibilities, relationships, or rights that define belonging to a nation. As GQ noted at the time, these episodes create more heat than light, fueling political brawls while overshadowing the lived realities of native people. The harm is not theoretical. When outsiders brandish DNA results as evidence of indigeneity, they sidestep the histories and treaties that have allowed native nations to survive against enormous odds. They also contribute to harmful myths that native identity is a matter of blood percentage or genetic markers rather than community and continuity. For the Cherokee, who endured forced removal, the trail of tears, and repeated attempts at erasure, the stakes are painfully clear. DNA as identity proof is not just scientifically shaky, it is politically dangerous. It shifts the authority to define who is Cherokee away from Cherokee law and into the hands of corporations or individuals. The backlash against DNA identity shortcuts also reflects deeper struggles tied to the freedmen. After the Civil War, formerly enslaved people who had lived in Cherokee communities became part of the nation. Their status has long been debated, entangled with ideas of race, federal policies, and later economic benefits tied to tribal citizenship. Wired has reported extensively on how these battles unfolded in courts and within the Cherokee Nation itself. Critics of DNA-based identity argue that introducing genetics only compounds the problem. If tribal membership were reduced to test results, it could erase the historical role of the freedmen, disregard treaty obligations, and reinforce racial divisions rather than heal them. For communities still grappling with legacies of slavery, removal, and assimilation, DNA shortcuts are not neutral, they are destabilizing. This is why ethical storytelling is so crucial. Time magazine has emphasized that narratives about Cherokee DNA must be handled with care. Sensational claims about dark secrets or hidden bloodlines might attract viewers, but they often drown out Cherokee voices working to protect language, culture, and sovereignty today. Elders are teaching the Cherokee syllabary to children, communities are reviving traditional ceremonies, and activists are fighting to preserve ancestral lands. These are the real battles for survival and identity, yet they can be eclipsed when the spotlight is placed on DNA anomalies or speculative connections to the old world. In the rush for headlines, the living story of resilience risks being silenced. The power struggle over DNA is, at its core, about who controls the narrative. Will Cherokee identity be defined by outside interpretations of genetic data, or by the Cherokee people themselves, who have endured centuries of attempts to take that control away? The backlash against DNA tests as identity proof is not just about science. It is about sovereignty, survival, and the right to tell one's own story. Strip away the headlines, and the question becomes sharper. What do we actually know right now, and what remains unknown? What we know and don't. After centuries of speculation and decades of genetic study, what do we actually know about Cherokee DNA? The most reliable answer is both clear and complicated. Mainstream genetics continues to affirm that the founding maternal lines of Native America are haplogroups A, B, C, and D, with haplogroup X recognized as a minor but important addition. PubMed-listed studies have shown again and again that these haplogroups explain nearly all native mitochondrial variation. While X once sparked transatlantic theories, researchers at California State University, East Bay, and elsewhere have demonstrated that it also appears in Siberia, meaning it can be accounted for within the long-established migration model. The claims that Cherokee DNA carries significant amounts of haplogroups T, U, J, or H as evidence of pre-Columbian Mediterranean ties remain unverified by peer-reviewed research. Far more convincingly, such markers are explained by well-documented historic admixture with European traders, settlers, and enslaved or freed Africans in the Southeast after the 1600s. That distinction is crucial. Science cannot confirm the sensational stories of Old World voyages shaping Cherokee DNA in prehistoric times. Instead, it points to something both less dramatic and more human: the reality that Cherokee communities, like many others, intermarried across cultural boundaries during centuries of upheaval and change. These unions left genetic echoes that persist today, but they do not overturn the story of indigenous origins rooted in Asia and North America. Even with newer tools, the limits remain stark. Autosomal DNA testing, which surveys the rest of the genome rather than just the maternal or paternal line, offers a broader view of ancestry. It can capture contributions from hundreds of ancestors across multiple generations. But as Teen Vogue highlighted, autosomal tests still cannot assign culture, citizenship, or community. Their results depend heavily on reference panels, which vary between companies and evolve as more data is added. A person tested today might receive a slightly different ethnicity estimate tomorrow, not because their DNA changed, but because the dataset behind the interpretation did. The shifting categories remind us that these tools are approximations, not definitive answers. They are useful for glimpsing population histories, not for declaring national identities. The Cherokee Nation, along with other native governments, has been consistent in its position. As PBS reported, DNA cannot and will not replace tribal law in determining citizenship. Sovereign nations set their own criteria, grounded in treaties, records, and community continuity. Genetics may illuminate aspects of the past, but it cannot substitute for the legal and cultural frameworks that sustain nations in the present. To ignore that boundary is to risk repeating old mistakes, where outsiders once again claim the power to define who counts as Cherokee. So the honest answer is nuanced. Some mysteries about Cherokee DNA endure, especially in how lineages diversified once they reached North America, and in the occasional presence of markers that seem, at first glance, out of place. But the best available evidence does not rewrite Cherokee origins. Instead, it reinforces what many have always known. The Cherokee are an indigenous people of North America, whose survival and identity are defined by law, memory, and lived nationhood, not by anomalies in genetic reports. And perhaps this is the real, darkest secret hidden in the bloodlines. Not that the Cherokee carry mysterious Old World ties, but that DNA itself cannot replace the stories, struggles, and sovereignty that define who they are. If the allure of genetic discovery tempts us to chase exotic answers, the deeper truth is more profound.
Nations live through their people, not their haplogroups. Like, comment, and subscribe if you want deep dives that balance bold questions with solid sources. Share your take. What should count more? Records and relationships, or lab reports? And watch the next video on screen for more untold stories that separate rumor from reality.